the very small fish. Of course, larger fish can get infected as well, but they do not suffer so much from the disease. But we can see extremely high mortality in rainbow trout fry in the hatcheries if they get infected with this Flavobacterium cycrophyllum. And that is the one which is actually giving rainbow trout fry syndrome or bacterial cold water disease. And therefore, we did a study if we could actually breed some more resistant fish. The problem with that disease is also that we cannot vaccinate the small fish because it often targets very small fry with an immune system which is not properly developed. So it cannot respond so well to a vaccine and get protected because the immune system is still very undeveloped in the very small fry here. So therefore, we need to find some other tool. And we have done some studies on that because then the question could be, can we actually breed some rainbow trout fry syndrome disease resistant fish? And I will show that to you now. Can I have the next slide, please? So therefore, we need to find some other tool. And yes, we did a challenge, several challenges, experiments in the same way. We produced the disease-free fish first in our rainbow trout hatchery, transported the very young fry to our infection facility, and then we exposed them to the Flavobacterium cycrophyllum, which is the pathogen giving the disease rainbow trout fry syndrome. And it will often develop quite fast and kill a huge part of the population in the trout hatcheries. And when we have done that, we selected, we checked all the fish which were surviving the infection, and we could see that on chromosome 25 on the rainbow trout genome, we actually see some genes which was conferring a resistance to the fish against rainbow trout fry syndrome. This is what, again, we call the Manhattan plot. Again, on the x-axis, we see the chromosomes of rainbow trout, 29 of them here. And then we see the frequency of the SNPs on the y-axis here. And you can see a huge amount of them associated with the resistance there on chromosome 25. Then we could go to the fish farmer, to the fish breeder, and say, please find some of your spawners you have DNA typed. Please pick out the ones which are carrying these markers and then make a new batch of eggs for us. And the breeder did that. AquaSearch over APS in Jutland, he did that. And then he transported the eggs to our hatchery. We could hatch out the eggs in the hatchery. We got the yolk sac larvae, and they swam up. And after a while, they were very still young, maybe less than 1 gram. We took them to our infection facility and exposed them to the Flavobacterium cycrophyllum again. Can I have the next slide, please? Yes, and here we could see that that the ones which were actually selected with the correct parents, they had a significantly lower mortality compared to the outbred fish. In the first trial here, we only used a limited amount of male spawners, but it was still significant. We could reduce the mortality of the population of the population in the outbred. We saw about 50% mortality in this case here, and then we could see that the QTL, the ones which were bred for resistance, they had a significantly lower mortality. But we could do better, actually, and we did that in the next trial, the second trial, which I will show you on the next slide, please. Yes, here actually the breeder made 4 genotypes, and that was based where he also selected female spawners with carrying the correct genes, which were carrying markers for resistance, and he used also male spawners carrying the correct genes and associated with resistance. And then again, he sent the egg batches to our hatchery. We hatched them out. We got the yolk sac larvae, we got the fry. We took them into the infection facility, and we exposed them in common garden setup as we usually do, where we mark the fish so we can tell the difference. And in this case, we saw actually a very high mortality of the two groups, which were had the low resistance, which actually did not carry the interesting genes here. They were selected because they did not carry those genes which were associated with resistance, and we saw up to 80% mortality in one group, and then the selected ones we had a very low mortality of about 20 to 25%. Of course, it's not absolute resistance, but we could see that we could actually confer some immunity, some natural immunities, some natural resistance to the fish by simply selecting the correct parents for this generation here. The reason we see some variation here is that there is also known that other genes are associated with this resistance against rainbow trout fried syndromes, but it was difficult in this situation to remove them or only target them because they were all present in the population of the breeders in all he had. In other cases, if we turn to other countries where they have other strains, we may find some which are more susceptible and some which may be more resistant. But these were the outcome from the rainbow trout fry syndrome experiment, where we could see we could also get some positive results, and we could validate the QTLs, the quantitative trait loci, and that gives also some idea that we in the future can find the similar things for other fish species and other diseases in other systems and other places in the world. Can I have the next slide, please? Yes, because now we have been talking about bacterial diseases, we have been talking about parasitic diseases, but we should also talk about viral diseases. And we have, of course, a lot of viral diseases also in salmonid fish, which we are working with. And I have to mention that colleagues in UK and Norway have done a great job working on Atlantic salmon. They have actually identified some interesting QTLs associated with a very strong resistance against the virus called IPN, infectious pancreatic necrosis virus, IPNV. And it has had an enormous importance for the production of Atlantic salmon in Europe. And their study was really interesting. I can show you on the next slide, please. Yeah, here we have from one paper by Thomas Moen from Aquagen in Norway. He published in Genetics in 2015 that they found one interesting locus where if you do not have the correct version of this gene in your genome, you will have a very low survival if you are exposed to the IPN virus. And if you have just one copy of it in your genome, your resistance will be increased significantly. But if you have actually 4 copies of it, you will almost be almost totally resistant to this viral infection. And that has had an enormous economical importance for farming in Norway, because IPN virus is a very serious disease in normal Atlantic salmon. So that is actually also employed commercially, where you can buy these interesting eggs here. So this is, but this fantastic work done by the colleagues in in UK and in Norway, they encouraged us also to look into, can we do something similar with our rainbow trout, because now we have the rainbow trout model, and can we also use that for viral diseases in rainbow trout? Can I have the next slide, please? Yes. So the question is, of course, is it possible to reach the same IPNV results for rainbow trout? Because IPNV is also serious in rainbow trout. But then I have to tell you that many groups have been working on that, and they are not as encouraging as we saw for Atlantic salmon. Um, we have to admit that even if rainbow trout and Atlantic salmon, they are related, they're in the same family, Salmonidae. There are actually differences when it's coming to immunity and disease resistance. And that goes not only for IPN virus, but we could also see that it's different when we talk about rainbow trout. So we were linking up with our colleagues from the Technical University of Denmark, who are normally doing challenges with viruses, and we produced some outbred rainbow trout from the AquaSearch, transported them to the infection facility, and then we followed the disease progression. And in this case, we did it several times, but we could actually identify, but not as strong as our colleagues in Atlantic salmon, that something is actually related to genes in the genome when we talk about resistance to IPN virus, also in rainbow trout. But they have been identified, and for the moment we are running the validation experiment, so I cannot tell you about the outcome of this. But again, there is still possibility to find a way out of the problems with the disease resistance and diseases in aquaculture when we are talking about viral diseases. Can I have the next slide, please? So I will not keep you here all day. I think you're already quite tired of listening to me, but I would just sum up and say I have a little take-home message here. So I think that my conclusion is, my experience is that marker-assisted selective breeding should be considered if you are producing fish in aquaculture. So I think it's a promising tool to elevate the health of aquacultured fish and secure sustainable farming. And that is actually a green solution. We are not using drugs for this. We are simply using our knowledge about genomics in this case. And we are not manipulating. We are not manipulating any genes here. We are simply selecting the ones which are behaving the best, are performing the best in aquaculture. So I think if we use this method here, the marker-assisted selective breeding, together with other approaches, which could be vaccination or better management and so on, I think we can increase the aquaculture producing around the world and still reduce the climate footprints from the production. So with these words, I would like to continue to the next slide. Can I have the next slide, please? Yeah, I just want to thank all my good colleagues who have been helping with all this work. I can tell you it's not so popular to stay in the lab during the night from midnight to 6 o'clock in the morning, from 6 o'clock to 12, from 12 to 6 o'clock in the evening, from 6 o'clock in the evening to midnight. But that was what we were doing during all these experiments here. And I could not do it alone. We needed a crew of good persons who have been taking part in all this work, a good teamwork. And we have published most of the work here. You can find them in these publications here. And we could not do it without some support from foundations and agencies as GODP and Innovation Fund Denmark. So thank you to all of them. And the next slide, please. Yes. So I would also like to thank you, the audience, for coming here today and listening to my talk, and especially to AKUATROP. who invited me and gave me opportunity to talk about these findings. And I hope that you in the future will can get some benefit from that. And those were the words from me today. Thank you very much for coming.
Thank you very much, Prof. Kurt, for that highly informative and thoughtful presentation. Your lecture has provided us with valuable insight and the principle of marker-assisted selection and its potential application in developing fish strains with improved resistance to disease. Ladies and gentlemen, we are now proceed with the question and answer sessions. You can ask questions and may submit it through the chat box. And please include your name and institution together with your questions. If you want to talk, you can open your microphone. We have one question here in our chat box, Prof. It's from Faiza from the Department of Fishery. And the questions are, did the antigen inside, oh no, her question is, may I know if you also take the fast growth traits into consideration during selection for disease resistant?
Is it the last question here? How can we confirm the hatchery production for fish and crustacean juveniles is 100% pathogen free? I mean, that was the first question here I should answer.
Yeah, we have the first question from Puan Faiza from Department of Fisheries, Malaysia.
Okay, should I start with number one? Did the antigen inside the vaccination?
Yeah, you can start from the number one, yeah.
Okay. Did the antigen inside the vaccination procedure have long-lasting effect in several generations of offspring? And how is anti-disease resistant rate over a several generation? Yes, I mean, when we do the vaccination, I mean, the vaccination, it will only confer immunity to the fish, the individual fish, which is actually vaccinated. Of course, there has been done a number of studies where we and other colleagues checked if for example, the specific antibodies produced in the fish were transferred in eggs to the next generation, and it could actually be seen that antibodies were transferred to the egg and could be detected. But for example, for rainbow trout fry syndrome, they gave no effect on the disease resistance in the next generation. And that was done, for example, with Scott LaPetra from the US and others. And so unfortunately, I cannot promise that you can confer immunity after vaccination to the next generation. I think it's a theoretical possibility because we can find antibodies in the eggs, yes. But I mean, the practical immunity in the next generation, I think that's it is not enough. Maybe only in the very early stages of development of the fish, you can get some protection from that. And then did the antigen vaccination be able to apply in invertebrates, crustaceans species? Do I have a different approach? Yeah, I mean, the immune system of crustaceans and invertebrates, it is different from what we find in fish. But the basic innate responses in some of the crustaceans, they actually are quite similar to what we also find in fish. And I'm sure that you can, it is not correct to say immunization and vaccination of crustaceans, but you can actually immunostimulate crustaceans and also other invertebrates to elevate their resistance to the pathogen. That has been done for snails and some crustaceans, but I mean, it is not the same approach as we use in fish vaccination, because in fish, we know that they produce antibodies, and antibodies are not produced by crustaceans and other invertebrates, but they have other ways to combat diseases. And that goes for lectins, that goes for complement factors, and so on. So there is a way to improve immunity by stimulating with antigens the crustaceans, absolutely. But it is not the same way it works in crustacean as it works in fish. So how did we confirm that hatchery production fish crustacean juvenile is 100% pathogen free? Yeah, I can only say that what we do here is that we have a general survey of all farms in Denmark where the Food Safety Agency and veterinary authorities go out into the farms, take samples at least once or twice a year, and they are checked for viral diseases. And that means that we, and also the ones which we know can be inside the eggs. And if you have a production facility as our breeder, in this case AquaSearch over APS, they are certified disease-free inside the eggs. Of course, you can then not be absolutely sure that you are free from diseases on the surface of the eggs, but then you disinfect the eggs before you transport them to a new place. And also when we get them in the hatchery, which has been disinfected, then we disinfect the eggs surface again, and then we are absolutely sure that we get disease-free fish. And we have checked that whenever we do these studies, we take out samples from the production and check if they are free from the different pathogens in the question. So this is the way we do. And I know it can be very difficult to do it in other systems. I'm sorry about that. And I can also tell you that it's extremely expensive for us. So it costs a lot of money to ask this production facility to produce those eggs. But this is the only way where we can be absolutely sure that we are free from them. And then there's a question here about growth. Yes, I can tell you that of course, our breeder, AquaSearch over, they have always have focus on growth. And they are incorporating growth trials all the time. So that means that they will not send us any fish without good traits for growth, because that's one of the basic performance in the hatchery to have spawners with genes associated with good growth. So they are working on it daily in the hatchery. So that is actually incorporated in the trials, that all the fish we have been using, they are actually growing well. So that's the answer to that question.
Any other questions from the floor? Prof, while we're waiting for other questions, I would like to ask you, between all the methods you mentioned before, which methods are the most effective as disease-resistant methods?
Yeah, it depends on which pathogen we are talking about, because, you know, when we talk about vibriosis, I mean, the main problem with the vibriosis is not in fry, it is in larger trout. And in that case, we can actually apply vaccination and confer very good immunity to the fish this way. But when we talk about the Flavobacterium cycrophyllum, which is a fry disease, this is where there we cannot vaccinate the fish because the immune system of the small fish is not developed enough to be able to respond effectively against the pathogen. So in this case, we have to rely on breeding and management and hygiene and stuff like that. And of course, hygiene is absolutely important. If you can actually keep the hatchery without those pathogens, it's absolutely the best. But otherwise, then selective breeding was a good way to obtain a better protection, because vaccination, I can tell you, we have vaccinated larger trout very effectively against Flavobacterium cycrophyllum, producing this rainbow trout fry syndrome. But you know, larger fish, they have a very good immune system. I mean, when the fish get up to 5 grams, it has a very good immune system, and you can vaccinate them against it. But that is not the problem in rainbow trout fry syndrome. The problem in rainbow trout fry syndrome is when they are very small, the small fish of maybe 1 gram, 2 grams, and the immune system in those are not sufficiently developed to give a very good protection. So we cannot have a good vaccination there. The larger ones, yes. And of course, I can tell you that when we vaccinate fish, the larger trout against rainbow trout fry syndrome, they gave a very good antibody response, very good response. And we can also see that some of these antibodies are transferred in the eggs to the next generation. But unfortunately, the protection is not good enough. They are not sufficient to give protection. So for the rainbow trout fry syndrome caused by Flavobacterium psychrophyllum, I really suggest that we should work more on the selective breeding, marker-assisted selective breeding, to get a more natural resistance in the fish against the disease. But again, vibriosis, vaccination is really good. Yersiniosis, vaccination is really good. About ick, for the moment, we don't have the vaccine. I hope we soon get one. But there we also have to rely on the marker-assisted selective breeding. So that was a short answer.
So what do you think? Do you agree if I said that all the methods you are saying just now also can be applied to the warm water or tropical fish?
Yes, I know that. Well, you know, I have not personally been doing those studies, but I know that Aquagen, the Norwegian company working on genetics, and others, they are working also on tropical fish, and they have also achieved very good results. And I know that a lot of other studies have been doing all around the world. So I think that it is one way that you also should look into in the tropical area. I really recommend that in the future. So yeah, that's the short answer.
We have questions here in the chat box from Dr. Professor Najia. For the mixed vaccination approach, is it immersion prime followed by injection booth?
Let me see. Yes, but you know, the reason, yeah, I mean, you could do that. I mean, you could do injection and then immersion. That's okay, because both types of immune stimulation will confer some immunity to the fish. But I just want to mention that the reason we recommend first immersion of rainbow trout fry, about 2 to 5 grams, where we see a good immune system in the fish, it gives a very good and very cheap protection against yersiniosis, and it will last about 6 months. And then we recommend injection, because then you do not need so much vaccine. You only apply 0.1 milliliter of the vaccine. But if you have very big fish, you know, the fish are growing within 6 months, very, very large, then you need a lot of antigens and to do immersion. So that's why when we have bigger fish, it's much cheaper to do the vaccination and applying much lower, smaller volumes of vaccine. So that's why I recommend that. And then there was one question about the new book on principles of fish immunology. Yeah, it was published by the Springer publishers in 2022, I think it was, yeah. And of course, if you enter the Springer website and search for it, this is the way you can achieve it. And I think it's also available as an ebook. So you don't need the printed form, but you can get it as an ebook as well. So that's absolutely possible.
Thank you, Prof. There's another question here. The question is, can you justify on combination of immersion and parenteral for vaccine delivery it the best in enhancing fish immunity?
Yes, I mean, I think that stimulation of the fish immune system, as we know from rainbow trout at least, it is that if you get a high concentration of antigen into contact with your immune system, as you do when you inject the vaccine into the body cavity or maybe into the muscle of the fish, then it will absolutely stimulate the immune system much better than if you just immerse the fish for 30 seconds into an antigen solution. Because when you do that, the antigen will be taken up by the gills and some of in the skin, and also some of the water with the antigen will be taken in as a drinking of the fish, and in the stomach and in the intestine, the antigen will be taken up and also stimulate the fish. But it is not as effective as when we inject the antigen into the peritoneal cavity, the body cavity of the fish. And then we have been doing also oral vaccination where we added the antigen into the feed. And then in that case, I can say that it was not as effective as the other trials we did with the immersion, bath, and injection. So I think when we use oral vaccination, it means that you put the antigens into the feed. You really have to have a high concentration, at least for rainbow trout, of the antigen. So we can be sure that enough of the antigen is taken up by the fish.
We have another question here. What fascinates you to conduct this kind of research?
Yes, I mean, I love fish, and I love fish. I have to say that we have more than 36,000 species of fish described in the world, and
Most of us
around
love fish.
and in aquaculture we have more than 500 species in culture. So that's great, and I love fish, and I love to see fresh fish. I love to see healthy fish. It is a big stimulation for me to see a well-behaving fish with a good performance and good reflections. But I get a little depressed when I see a diseased fish. So if we can improve the well-being, welfare, and health of fish, it is a gift to me. Yeah.
Last but not least, where do you think these methods, this selective breeding can go? How far does it can go in aquaculture?
Well, I think it can be applied in all species in aquaculture. I think, and that goes not only for fish, but also for invertebrates, I mean crustaceans. I did not work on that in the crustaceans at all, so I will not come out with any details about that. But I'm sure that in all cases, the basic principles of the disease resistance may be more or less the same. So I think that in the future, I can also encourage people to work on invertebrates, including crustaceans, and do selective breeding. But I will not be the expert in crustacean biology. I can only talk about the fish.
Thank you so much, Prof. Kurt. I think, ladies and gentlemen, that concludes our question and answer session. We apologize if there's any question that I missed to read. So due to the time constraints, we are unable to address every question received. So as today's presentation, we can highlight that several important points, which is marker-assisted selections allow genetic information to be incorporated into selective breeding decisions. Genetic markers associated with disease resistance may assist breeding program in identifying suitable broodstock more efficiently. And disease-resistant fish strains may improve survival production, efficiency, and farm profitability while reducing loss caused by infectious disease. The knowledge shared today demonstrates that selective breeding and genetic technologies can make an important contribution to more resilient, sustainable, and productive aquaculture system. Ladies and gentlemen, on behalf of the Institute of Tropical Aquaculture and Fisheries University Malaysia Terengganu, I would like to express our sincere appreciation to Professor Dr. Kurt Buchmann for sharing his valuable knowledge, expertise, and experience with us. Thank you, Prof, for accepting our invitation and for contributing to this international knowledge sharing program. I would like to invite our director, Professor Chemist Technologist Dr. Nur Azman Kasan, to give some words. Please welcome, Prof.
Okay, Assalamualaikum warahmatullahi taala wabarakatuh, and very good afternoon. Prof. Kurt, how are you? All the way from Denmark.
Thank you.
Thank you so much. On behalf of AKUATROP and also UMT, we are more than happy to see you online, and we hope to see you in physicals somewhere in September, Prof. We are still arranging for that. We hope that I noticed that there is some more question that been raised by the participant, but maybe we can keep it, all right, if we have time. But if we want to see Prof. Kurt in person, we hope that you can join us somewhere, I think in the middle of September. On behalf again, Prof, and I would like to take this opportunity to appreciate and also wish everyone who participate in today's talk. And I've been informed that for today's we have like more than hundreds of participants all around the world. And also like to welcome and also thank you to our counterpart from the Department of Fisheries, from the University of Putra Malaysia, and other. I didn't receive the whole participant list, but I would like to say thank you so much for attending our workshop or physical workshop. I don't know, online workshop today. So without further ado, again, Prof, we hope to see you again in 2 months' time. All right, in person. Hope to see you and safe travel to Malaysia. For all the participants, for your information, Prof. Kurt Buchmann is our visiting professor, and we hope to see you again in 2 months' time, inshallah. Okay, with that, I get back to Puan Yati. Thank you, Puan Yati. Masih.
Thank you, Prof. Dr.
Thank you.
Azman. Ladies and gentlemen, before we conclude, AKUATROP is pleased to announce its upcoming program on workshop on basic aquaculture parasitology 2026. You can access the information on our chat box, and we also have put the register link there. So this workshop will be held on 27 September 2026 at AKUATROP, University of Malaysia, Terengganu, and features, of course, our speakers today, Professor Dr. Kurt, and we provide participants with fundamental knowledge and practical exposure related to parasite affecting aquaculture species. For the information regarding registration, fees, and activities, you can contact our official AKUATROP office. We warmly invite researchers, students, technical officers, aquaculture practitioners, and other interested participants to join this workshop. And ladies and gentlemen, AKUATROP would also like to thank the organizing committee, technical team, and supporting staff who have contributed to the successful organization of this program. Thank you so much. And we are lucky to have almost 82 participants today. And our sincere appreciation is also extended to all participants for your attendance, questions, and active participation. And before we ended this online workshop, I would like to ask everyone to open your camera for us to take a screenshot pictures. So please open your camera before so that our technical team could take screenshot pictures. And thank you again to Prof. Dr. Kurt Buchmann and to everyone who joined us today. Are we ready?
Can we try to adjust the photo?
Okay, we still have some who still unopened the camera.
Okay. Okay.
sini. Nambahin mo banyak kan? Yang bawah banyak. Saya start streaming